Good afternoon. Welcome back to our final breakout session. I'm Des Kelly, a developmental behavioral pediatrician and chair of the board of the Institute for Child Success. And I'm glad to see you again this afternoon. And thank you for joining us. And will be the Genetics of Autism breakout session. We're looking forward to a great discussion and presentation by Dr. Rogers. Just a few logistics. As I think you've heard this in your previous breakout sessions, but just to be sure, the session is being recorded and the chat is being archived. You're in view-only mode, so please enter any questions or comments through the chat box. And also, if you're having any technical issues, please do that. Share there and we'll get help for you. The session, as you know, I think uses auto captioning. So if you click the live transcript button at the central bottom of your screen, that'll get you to the auto captioning. And just a quick note again on professional credits, development credits. I'm going to put in the chat box right now the links to the SC Endeavors site for early educators. If you could link to there and complete the submission form before the end of the session, that'll enable you getting credits. For CME, we'll be sending you an email in next week with more specifics about CME credit. Any questions in that regard, I do have John Conklin's email address in the chat. So it's jconklin@instituteforchildsuccess.org. We have heard that some folk have had difficulty with the SC Endeavors credits on Google or in Gmail. So if you are running into difficulty, email John and he'll help you resolve those issues. And I think that covers the housekeeping. The session is eligible for CME credits as well as we've noted. So at this time, it's my great pleasure to introduce Dr. Curtis Rogers, who is just a great guy. We've been colleagues for many years here in Greenville. Dr. Rogers is director of the Greenville office of the Greenwood Genetics Center and is responsible for clinical genetic services in the upstate of South Carolina. And he founded and has led that office up here for over 30 years and does where they do provide a broad range of genetic services, clinical evaluations, particularly infants, children, adults with birth defects and intellectual disabilities, storage disorders, neuromuscular disorders, counseling prenatally, preconceptional. Dr. Rogers is an expert. He has a special interest in the etiology and clinical aspects of skeletal dysplasias and craniofacial anomalies and has published extensively on that. And he was also on the team that first reported on Phelan-McDermott syndrome. And Dr. Rogers has done a lot internationally with those patients and families. He, as I say, is a highly respected colleague. He's somebody who goes out of his way in his dedication to his patients. From Down Syndrome Foundation, I could go on and on, all of the work that Dr. Rogers does, both as an expert clinician, as a researcher, as a supporter of families and children. You're not going to find anybody who knows more about the genetics of autism than Dr. Rogers. I'll stop there before I use up the whole time and hand over to Dr. Rogers. And thank you very much for being with us.
Thank you very much, Des. You will find someone who knows more about autism than me, I promise you. Unfortunately, there are a lot of gaps in our knowledge about the genetic etiology of autism, and we will get into some of that. So I'm going to get started here. It's Friday afternoon. I know we all want to have something else to do later today. So, from a parent's perspective, and I think whenever we talk about things, whenever I learn something, whenever I discuss something, I always want to look at it from the parent or the patient's perspective because that's what matters. Uh, from a parent's perspective, they want to know why did this happen? In this case, we're talking about autism. Will it happen again? So we'll mention a little bit about recurrence risk. Are there things I can do to keep it from happening again? What does the long-term outlook or prognosis uh, look like for my child? Are there other associated problems that I need to be watching for? I think importantly, and where we all come into here, and certainly as a team, what supports and services are available for that family and what can they do to access those services. And I can tell you when we see patients and families in our offices, sometimes that's not the easiest thing to do is access services. So we rely a lot on BabyNet, the Department of Disabilities, Special Needs, Family Connections, things like that to help our parents access services for their child. And is there a treatment? And I'd love to say we have specific treatments, but I happen to be in a field of genetics where we do not have many specific treatments. We are getting much better. We're developing these, but nonetheless, we still have a long, long way to go before we are ready to say we have a treatment for many genetic disorders. Right now, our main specific treatments are geared toward metabolic disorders typically for those seem to be the easiest to treat by being able to replace a protein which the body is missing because there's an error in the gene that codes for that protein. The genetics of autism is confusing and confounding because the criteria keep changing. I don't have anything to do with that, but I get affected by that. There's an increasing prevalence. Part of that may be due to the changing criteria. As far as the etiology, we certainly have a long, long way to go and many roads to travel. Right now, we'll talk about the testing that we have, and what we have is what we have, but that doesn't mean 2 years or 5 years down the road, we won't have a new test. Treatment. I think our problem with treating with autism is that it is a very widely variable spectrum with an incredible number of causes, and that makes specific treatment difficult. And I think many times we don't even know in the brain exactly what we're treating. Is there, we'll talk about this later, is there too much excitation? Is there too much inhibition? Vice versa. What are we treating and how do we get about treating that? And I think we also have to worry with treatment. Are we going to get any off-site effects or adverse reactions? Because as we start talking about treatment, I'm going to mention a couple of things to you where imbalance in the expression of a gene in either direction can lead to a genetic disorder associated with autism, intellectual disability, whatever. So when we're trying to tweak, let's say, the expression of a gene and its protein in the brain, we have to be very careful about how we do that. We'll talk a little bit about research and then conclude with some frustrations that all of us have when we talk about autism. So remember, highly variable. That's why we call it a spectrum. There is, depending on what you read, a difference in heritability and certainly in penetrance. Heritability implies that what is the risk in general that the condition we're discussing, in this case autism, has a genetic component and how strong is that genetic component. Penetrance, on the other hand, is if you have that genetic component in your family, what is the likelihood that you are going to express that in having autism? Heterogeneity refers to the multiple genetic and environmental factors that combine to contribute to the causation of autism. I will tell you there's a lot we still don't know, and there is no holy grail gene. If there were, we would have found it by now, but it is not there. And so what you're going to see as we discuss this is there's a dual train of thought here. One, that you have a combination of changes in many different genes that additively or synergistically, along with environmental factors, contribute to that picture that we call autism. Or there may be some very strong, think of them as stronger genes, that in and of themselves, an alteration in that gene appears to be sufficient to cause a high risk of autism. Here you can see a little bit of a history from a fellow in France, Thomas Bourgeron, who plotted this out. What you can see on the bottom is a karyotype in green, microarrays, which we commonly do now, whole exome and whole genome sequencing. You can see the rate of discovery of genes has increased rapidly. And you can also note that the prevalence of ASD, when you look at that left-hand y-axis, has dramatically increased too. So we found more genes, and at the same time, our prevalence of ASD has been increasing. I'm not sure we've made much of a dent in improving our ability to find the cause. We're getting better. If you look along the x-axis here, what you will see is some of the early genes, Fragile X, FMR1, TSC, tuberous sclerosis. There's also a TSC1 gene. You look above TSC1 gene right there at 1997, you'll see UBE3A, which is the gene for Angelman syndrome. Right beside that, METP2 for Rett syndrome. And it goes on and on. What you'll see is more and more genes have been found which have been identified in association with an increased risk of autism. This is not to say that everyone who has a pathogenic variant in one of these genes is going to have autism. We'll take SHANK3. If you go to 2007 there, you'll see SHANK3, which is the main causative gene associated with Phelan-McDermid syndrome. We certainly know that there are individuals who have alterations in that gene, point alterations in that gene, and deletions of the entire gene that do not meet the criteria for a diagnosis of autism. So there are a lot of difficult things here as far as trying to figure out what causes autism. Many times I think it's easier to figure out what has not caused autism in that particular person. And many times that's what we're doing. We're ruling out things. So I mentioned epidemiology, the increased prevalence of autism. Part of that may be heightened awareness. I know we've talked about this at meetings before, the broadened diagnostic criteria. And then some people have implied that, well, maybe this rate of autism is going up in part due to increasing environmental influences from pollutants, let's say, in our environment. You can see that as of 2016, and I can't keep up with the numbers, but 1 in 54 children has been estimated to have autism, and a third of those at least have associated intellectual disability. I would tell you in genetics, we have a much better hit rate or discovery rate for a specific etiology. when there are associated features like intellectual disability, probably because we know more about the genes associated with intellectual disability. One common thing that has remained, no matter how much that rate or incidence of autism has increased, there have always been a significant increase in males compared to females with autism. And that number has stayed a constant for roughly 4 times as many males as females. And you would think with all we know about the X chromosome, we would have figured that out, but we don't know. Just a little jab at you guys who make up all these criteria. It keeps going and going and going. And as the criteria change, it seems the rate always goes up. This is the definition, I think, that we find in our genetics literature: a broad group of neurodevelopmental disorders characterized by young age of onset, impairment of communication and social abilities, restricted interest in repetitive behaviors, and symptoms that affect patients' function in various areas, and I'm going to say of their daily life. The other thing is that symptom severity varies widely and may be associated with compounding comorbidities. Intellectual disability is the big one many times that we see. I certainly believe in that gut-brain connection. However, I don't understand that very well. I pulled some of these slides from a talk I had on intellectual disability, and I think they apply to autism as well. I think they apply to any complex neurodevelopmental problem because our brain is so complex. And it is true that intellectual disability and autism are primarily due to deficiencies in neuronal network connectivity. Now, that can be due to a number of different reasons, and we'll talk a little bit about those. Remember, the main things that you have to do in your brain is you have to first make brain cells or have neurogenesis. The second thing is you have to have appropriate neuronal migration, which is a problem in and of itself, getting those brain cells to the right layer in the brain and to the right region of the brain. And once those cells get there, ensuring that there is appropriate synaptic connectivity that occurs in both the excitatory and inhibitory synapses. It is a complicated process. I always say, it's a wonder any of us can walk and talk. It is highly diverse as far as the disorder of autism or intellectual disability, and there are multiple underlying causes. In genetics, one of the things we talk about is multifactorial disorders. That's to make us sound like we know more than we do, because then we can say, oh, it's genetic and environmental factors lumped together. We certainly know for autism that there's a very strong genetic component. I'm sure all of you have read twin studies where they compare monozygotic and dizygotic twins to each other, and twins that were raised in different environments. And then you come back and you look at, well, how many had, in this case, autism out of this group? And we can calculate a heritability factor or relative risk in siblings based on that. I will tell you that no twins are identical, because there are always going to be mitochondrial and epigenetic differences that at this point we certainly have no way to account for. There is going to be an increased risk with numerous gene variants, and most of the families we see, and I'm sure you've seen families with 5 children with autism spectrum disorder, and we have studied that family top to bottom, left to right, and we have not been able to put our finger on one specific mechanism or gene or environmental factor in that family. Obviously, there's a strong genetic component in that family. We are just not yet smart enough to identify. It's been estimated that there's an underlying genetic cause in at least, or can be identified in at least 10 to 40%. We're getting better. I think we will continue to get better. And I think once we are able to figure out how to account for multiple changes in different genes, then I think we will become even better at understanding the etiology of autism and possibly better at providing specific treatments. I mentioned to you that if you have a positive family history, intellectual disability, or abnormal physical features, or birth defects, we are much more likely to be able to pinpoint a specific diagnosis. We've got more to hang our hat on. We fancy ourselves to be Sherlock Holmes, and we need more clues, and that provides us with those clues. There's certainly an increased recurrence risk in families who have one or more affected children. And we get into this all the time with families. If you have one affected child, the risk in the full siblings of that child is 4 to 7% in a subsequent pregnancy. And if you have 2 or more affected children, that risk jumps dramatically and can be as high as 1/3 in the next pregnancy. There's a much higher concordance rate between monozygotic versus dizygotic twins, which makes perfect sense because we feel there is a high genetic load, and overall the heritability has been estimated at 90%. Remember that the difference between monozygotic twins and dizygotic twins, monozygotic meaning coming from one fertilized egg or zygote, so that zygote then splits apart into 2 separate zygotes, so that they should, quote, share all of their genetic material. However, that is ignoring the fact of those mitochondria and the fact that epigenetic changes can occur during and after pregnancy. Dizygotic twins are no more closely, that's 2 eggs fertilized by 2 different sperm. They are no more closely related than my brother and I are, and he is 3 years older than me. Dizygotic twins just happen to be womb mates for 9 months. That was our joke for the day. We also have a better chance of finding a specific etiology for autism or intellectual disability if there is a recognizable pattern. I mentioned, well, gee, we'd like to have some other features to help us. A syndrome is a recognizable pattern of malformations and neurocognitive findings that implies a single diagnosis. So that gives us something to hang our hat on. We can look at that face, we can look at the brain findings on an MRI, and that may lead us to a specific diagnosis. Those non-syndromic cases are much, much harder to diagnose, but in intellectual disability and autism, the non-syndromic individuals make up probably 2/3 to 3/4 of the individuals that we see with ID or ASD. We are finding now that there are many genes that can cause both syndromic and non-syndromic intellectual disability or autism. And so that division is becoming blurred, but that's okay for us. We just say we're expanding the phenotype that we can see with that particular gene and change it, pathogenic alterations in that gene. There are some kind of little phenotypic hints that we look for. we have a better hit rate for finding the cause of autism in individuals who have macrocephaly or a big head greater than the 97th percentile. Microcephaly, not so much, because seems to be there are a lot more things that can go wrong to cause you to have microcephaly than macrocephaly. I mentioned intellectual disability gives us a leg up in finding a cause, as do seizures, because then we start looking for calcium channel genes and things like that, or other channelopathies that may lead us to a diagnosis. Remember, we're also, when we're finding changes in genes, we're having to correlate the gene and its physiologic function with what we are seeing phenotypically in the patient, and quite frankly, trying to see, does this make sense that a change in this gene is causing what we're seeing in this particular patient, or in a group of patients, if we've been lucky enough to get a group of patients. And we have ways to do that now. There's a thing called Gene Matcher, where we can find rare variants and genes that have not been reported or very infrequently reported. We can put them out in this Gene Matcher database, and people from all around the world will start responding. Yes, we saw a change in that gene, and our patient had similar phenotypic features to your patient. Maybe we're on to something. So we have a lot of tools at our disposal that are helping us rapidly increase our knowledge. I will tell you there are hundreds of genes and loci implicated in autism, and that is not surprising. If we look at the synapse and think of the billions of synapses you have in your brain, or trillions, at each little tiny synapse, there are over 1,500 genes and corresponding proteins which they encode that function at every one of those tiny little synapses. It is quite complex. That's why it is extremely heterogeneous as far as its cause. I will say in research, our problem, and I think I mentioned this again at the end, our problem is patient selection. Autism, the description of autism is not specific enough for what we need when we start looking for genetic etiologies. It's not sufficient to help us rule out a lot of things. So this careful patient stratification is important, and how those are stratified behaviorally, or possibly a stratified because of a structural anomaly or specific behavioral phenotype, becomes incredibly important for us when we are trying to, one, delineate a cause, and even more critical when we are trying to treat patients and trying to come up with a treatment that in general will be useful for a certain group of patients. And I will tell you that certain group of patients doesn't have to have the same specific gene involved. I think what we're going to find is much like when we treat infections, we're going to develop some anti-autism drugs that will treat a variety of causes. We don't need to know whether it's strep or staph or whatever is causing this infection. We are identifying it as an infection, or autism in this case, and then we're going to be able to have some groups for which we will have more specific treatments. We know there are a lot of neuroanatomical abnormalities in ASD. Thus far, we have not been really very good at using these to differentiate patients. I think in the long run, we'll get better imaging. We'll get better spectral studies that will show us how certain drugs affect certain areas of the brain. And I think that will lead to better treatments. So when we look at the etiology, you can see that there are an incredible number of factors. Copy number variation, in other words, extra or missing pieces of a chromosome or an entire extra chromosome. Epigenetics, which are non-structural, they are not altering the structure of the DNA, but you can think of them as nature's way of turning on and turning off genes, and we may be inappropriately turning on and turning off genes at a time when they do not need to be off or on, and that can go either way. Environmental factors that may contribute to epigenetic changes, double hit mutations or variants where you have to knock out both copies of the gene, or in one instance, you may have genes that work in the same pathway that are on the same chromosome. And if you happen to have a variant in both of those genes, you may alter that pathway function. And we certainly don't know much about hormones and sex-linked modifiers yet, but we think there must be a role there since we have 4 times as many males as females affected. Genetic susceptibility means that while you are at a higher risk for having autism, having a change in many of these genes does not mean you are absolutely going to have autism. And in fact, you may not have any neurodevelopmental issues at all. And if you do have neurodevelopmental issues, they may be highly variable. The genetic architecture is exceedingly heterogeneous, and again leads us back to the problem. And I mentioned this at the start. It can be complex with a huge burden of low-risk variants that are additive or synergistic with each other to lead to that autism phenotype, or we can have highly penetrant, strong, rare variants. Think Fragile X syndrome, think Shank3 and Phelan-McDermid syndrome, MECP2 and Rett syndrome. Those types of genes for which we find a low but significant percentage of individuals with autism having an alteration in one of those genes. And we can never forget the heterogeneity of the genetic background. When we're talking about 1 gene or 10 genes or 100 genes, remember there are over 20,000 different genes in each cell of your body that may be playing a role in every function of your body, including your brain function. So we cannot leave out those other genes and say that they're not important in the pattern that we're seeing in a specific patient. Common SNPs are distributed, or single nucleotide polymorphisms are distributed all over the genome. They've been estimated to account for at least 20% of autism spectrum liability. And again, act additively or synergistically by themselves. Each one of these changes probably would cause no issues, but it's very complicated. The other thing is you'll see that many neurobehavioral disorders are caused by the same genes, schizophrenia and intellectual disability. So what is the connection there? I think part of that is the brain has only so many ways it can respond to dysfunction as far as what we see externally in a phenotype. I think I've covered a lot of this. the increased rate of de novo variants is much higher in autism spectrum disorder individuals in a family than in their unaffected sibling members. Again, telling us that these copy number variants, small extra or missing pieces of chromosomes, are more common in the individuals who have autism, even within a family. There's a clue for us. And that loss of function, mutations, or pathogenic variants are more likely to occur de novo or new, 2 times more commonly in the children with autism spectrum disorder in a family than the children who don't have autism in that family. The other thing we have to be concerned about, and we're learning more about, is somatic mosaicism, meaning that is not present in all your cells. That variant may be only present in a portion of your cells, occurred post-zygotically, that was after the egg and sperm came together, and they may account for some of those changes. And then you throw in the epigenetic changes on top of that, and you can see how difficult this becomes. The epigenetic changes, again, don't structurally alter the DNA, but lead to typically histone changes, methylation changes that either repress or promote gene activity. And again, our genes are turned off in a very controlled fashion, in a temporal fashion, and altering that can lead to dysfunction and even malformation or improper formation of brain cells and synaptic function. Some people have felt that dysregulated methylation of genes on the X chromosome, and let me tell you, genes are packed in on the X chromosome. It is a very important chromosome that dysregulated methylation of the genes on the X chromosome may be a risk factor for autism, but also may be a significant risk factor for the male to female predominance in autism. Let's look a little bit at a synapse here. All of these genes, I won't say all, but many of these genes, a majority of these genes converge on synaptic function and brain connectivity. And if you just look at this simple diagram of a synapse, I can tell you that orexin, neuroligin, SHANK3, UBE3A, the mTOR genes, FMRP, TSC1 and 2, all of these genes are associated with autism and can be associated with intellectual disability as well, and function either at the presynaptic or many of them at the postsynaptic density, which is shown at the bottom of that little cartoon there with the receptors on. You're seeing the mGlu receptor for the glutamate granules that you see floating in that synaptic cleft, and stimulating those leads to increased protein production and signaling in other neurons. So a lot of different genes there, a lot of different genetic syndromes, and certainly a lot of individuals who have non-syndromic autism have changes in genes associated with the synapse. However, no single molecular cause or gene accounts for more than 1 to 2% of autism spectrum disorder cases. Man, that's depressing when I think about that. Again, there is no holy grail. We didn't find that gene we were looking for, the autism gene. What we found were a bunch of genes. As I mentioned, if we can identify a syndrome, at least we have a target to shoot for when we start testing genes. However, new technologies have allowed us to expand our testing so that we don't have to be as good a shot these days with our targeted testing. We don't even do targeted testing many times now. We will go straight to either whole exome sequencing or whole genome sequencing to look for genetic abnormalities. You can see that karyotypic abnormalities, chromosome abnormalities, copy number variants lead to 5 to 10% of autism, and then look down the rest of that list after Fragile X syndrome being 2%, and tuberous sclerosis being 1%. Boy, that list drops down. There's my friend Shank 3 and at 22q13 with Phelan-McDermid syndrome. And again, a very, very low hit rate when you go looking for a specific gene. Rett syndrome, 0.1%. And these numbers are going to vary depending on which study you look at. However, the numbers are always very, very low. I mentioned the cytogenetic abnormalities. If there are dysmorphic features in the patient or intellectual disability, we're more likely to find a specific cytogenetic abnormality, and you can see that greater than 450 different copy number variants have been associated with ASD. So we can do routine testing. It's kind of an old hat now. We don't typically do that unless we're looking at a child who has Down syndrome clinically. We might do a chromosome study because it's the quickest thing and cheapest thing to do. We can do FISH testing, but nowadays, most of us are going to do microarray testing. You're going to hear me mention whole exome sequencing. After that, you can do whole genome sequencing, which you can think of as a way to combine whole exome sequencing and make it even better, as well as combine that with a microarray. So whole genome sequencing is the promise of the future to look for both molecular variants and copy number variants. But we still do a lot of microarrays, and we still do a lot of whole exome sequencing. So just to show you what a karyotype looks like. And back in 2010, this was recommended as a first-tier test for ASD, and it is probably still recommended as that by some insurance companies. So we would like to, many times we're hamstrung a little bit, so we're going to go to a microarray first, and typically after that, unless we have a few specific genes in mind, then we're going to get our microarray back. Then we will go to molecular testing. You can see some of the copy number variants that we see. Chromosome 15 and 16 are hotspots as far as copy number variants in autism, other behavioral problems, and intellectual disability. And that deletion down there that involves SHANK3 is the deletion that causes Phelan-McDermid syndrome. You can see the percent who have autism spectrum disorder that have that particular, that if you have this particular copy number variant, for instance, 22q13, greater than 50% have autism. But if you look at it from the other side, and you're looking at a large population with autism, only 1% or less of them would have a deletion of 22q13.3. Here are a bunch of kids who have 16p11.2 deletions. I told you this was a hot spot. I look at those kids and I say, to me, there's absolutely no unifying physical features that I'm going to identify on their face. And therefore, that's why we do this microarray testing to try to delineate a specific etiology. You can see on this slide, and I know it's busy, but the reason for showing you this is there are many, many rare genes that have an unknown contribution to autism, or if you have a change in that gene, we don't know what the rate of autism is in individuals carrying that gene, but all of these genes listed on here have been associated with autism spectrum disorder in at least one individual in which this gene change has been reported. So again, it is a hard search to do. Some of the more common ones in which we find single gene changes: Fragile X syndrome, Rett syndrome with MECP2, and tuberous sclerosis and Phelan-McDermid syndrome. The reason why you wonder why we keep using those is because they're the most common. They're the ones we're going to have the highest hit rate for, and the ones therefore that we have the most experience with. Here's a young boy and an older adult with Fragile X syndrome, and depending on who's doing the testing, 15 to 25% of them would have autism. Can be very, very difficult to recognize in a young child based on phenotypic and neurobehavioral features. And Fragile X was called that because of this little fragile site at the bottom of the X chromosome. We now know it's due to an expansion of CGG repeats, and that leads to methylation, which silences gene expression. So we turned off that gene. And you'll see what that gene does in a little bit. A lot of different single gene abnormalities and different genes that have been associated with autism. I mentioned a couple of these. P10, I talked about macrocephaly. This is one of the genes, PTEN, and PTEN that can be associated with autism and macrocephaly. So that's one of the first ones we think about. We can use Sanger sequencing, which is the gold standard, but it's an old way of doing it. It's fine if you're looking for one specific gene, but now we can rapidly sequence the entire exome and genome using next generation sequencing that is done automatically for us. And it's been quite the discovery in genetics. So multi-gene panels we use. Some panels are better than others. Not all panels are the same. They may have the same genes, more genes, less genes. I really don't use panels that much anymore unless I have a very specific group of disorders that I'm looking for, because whole exome sequencing or whole genome sequencing are the way to go these days. You get more bang for the buck. Remember that exome sequencing is sequencing the protein coding parts or the exons of the genes, plus a little bit on each side of the exon into the splice site of the intron, whereas whole genome sequencing is sequencing 3 billion base pairs, one cell's worth of DNA, and by doing that, you hope to find more and learn more. Autism gene panels in our past have certainly been helpful, but nowadays we don't use those much anymore. You're going to see some rare genes here associated with speech delay and autism and things like that. Other individuals with even rarer genes that have been associated with autism. So let me rip through the rest of these right quick, because I don't want to keep you guys all Friday afternoon. But there is no treatment currently available for a specific etiology of autism. Not that I know of. And we have tried a lot of different things. And a lot of these are general treatments that have been tried. And you can see the number of publications has increased dramatically, which is what this graph is showing, for these different methods of trying to treat. You can see some of these alternative treatments. We do not have any that I know of specific effective pharmacologic treatment, which gets us back to trying to put patients with autism spectrum disorder into functional groups that may allow us to develop a treatment for a larger functional group. Not all patients with autism. However, maybe there are a group of patients who will respond better to a particular drug than will another group. And again, this comes to how do you segregate the groups in order to do pharmacologic testing on a group so that you get the best results that you can and are more likely to get a positive response to a drug. So these two guys, Rubenstein and Brzezinski, came up with this idea of excitatory inhibitory balance. And this makes sense to me as an engineer and a physician. And so I like to look at things this way as far as an imbalance causing the autism or dysfunction of the brain. Could be intellectual disability, it could be schizophrenia. There, just to use one pathway, the wingless pathway, we have two things going on here, and I'll show you the pathway in a second. But beta-catenin is needed for cell adhesion and cell signaling. You lose function of that, you have decreased wingless or Wnt signaling, you wind up with a patient who has microcephaly and ID. But guess what? There's a gene called CHD8 that codes for a regulatory factor that directly inhibits beta-catenin. And so we have this balance going on here. You're seeing this coming into play. Loss of function in CHD8 results in increased expression in the Wnt pathway signaling, and you get somewhat the opposite in the fact that you get an individual with macrocephaly and ASD. So very interesting. MECP2 is very similar that way because imbalances of MECP2 in that very tightly regulated gene and its expression can lead, if there's too much MECP2 expression or too little MECP2 expression, you can wind up with severe intellectual disability and autism both ways. So here you see CHD8 with a stop sign around it, trying to put the brakes on BCAT. And BCAT is trying to help with cell connection. What you see over there is lithium acting as a stop or inhibitory mechanism that leads to less breakdown of BCAT. And so the idea is that lithium allows you to express more beta-catenin, and by doing that, that leads to better brain function. However, we know that has to be tightly balanced. So another pathway, the retinoic acid pathway, in which UBE3A, the gene associated with Angelman syndrome, if you have a duplication of 15q, which includes UBE3A, you get autism. But if you have deficiency of UBE3A, guess what? You get Angelman, and you can also have autism. So a lot of different things there, and this tight balance in your brain, which again makes me wonder how any of us can walk and talk because of the incredible complexity and regulation of the pathways that allow us to think. Back to Fragile X syndrome, again, you see it functioning here in the postsynaptic density right in the middle of that cartoon, FMRP. And what it is trying to do is decrease or dampen down increased protein production. And if that does not happen, you get imbalance with overexcitation at the synapse. So the idea is that FMRP, when normally functioning, will dampen that down. And when it is not present due to Fragile X syndrome, that protein is not made, then you have too much excitation, too much protein production, and that leads to that altered excitatory inhibitory balance. Here are the medications that have been tried and where they would function in the synapse to try to dampen down that hyperactive system. And I think you can see here that one is incredibly complex. And I'll tell you, two, none of these medications have been significantly successful in the Fragile X model and even in Fragile X patients in providing what we would consider a good treatment for Fragile X syndrome. So one that people talk about is arbaclofen, and you can see arbaclofen is over on the GABA side. So what you're trying to do is it's an agonist, so you're trying to increase the inhibitory synaptic function to counterbalance the increased excitatory function caused by the loss of FMRP in Fragile X syndrome. But thus far, we don't have a great clue there, and we've been working on this for a long, long time. So I'll skip that one and just tell you we're not there yet. So in conclusion, we have heightened awareness, changing definition that leads to a prevalence of increasing ASDs. Who knows, there may be environmental fluid influences playing a role. We certainly know there are many different genes involved, and it may be a combination of genes that are leading to autism. My kind of thing is we need to develop for families, I think, and also I think for seeing who may respond best to medications, this autism risk molecular score. That's my little term I made up, because I think when we get to that point, we may be able to better quantitate response to different medication trials and what may work for who. The other key thing is we need definitive biomarkers, whether that be a blood test, an MRI, whatever, to be able to segregate patients into groups that have autism and be able to monitor response to treatment for those individuals. So we need to take the parents out of the response equation. I love parents, but I also realize the placebo effect that occurs, because if you're a parent and your child has a health issue, be it autism or whatever, you want them to improve when they take a medication. And that is difficult to engineer out of a study when we do that. So we need better objective measures, and maybe those biomarkers come into play there. And we need better identification and understanding of the common pathways that can cause autism, so that we may direct therapy toward those particular areas. And I think that's the last slide. So I will stop sharing, and I will be glad to take any questions anybody might have.
So thank you very, very much.
That is a mouthful there, trying to talk about the genetics of autism. I see some questions coming up.
Yes.
What do I think about the thoughts on the constantly changing criteria for genetics and autism? I think if you say they have autism, I'm fine with that. I think the problem is not who we say has autism, it's how we divide those individuals with ASDs into specific groups that we can more carefully and appropriately study. So, the incidence, the changing incidence doesn't do anything to us in genetics. We just want a better way to segregate into groups, and I'm sure everybody does. Then another question was, are there particular regions of the brain that are affected, or do the genetic synaptic changes have more of a global effect? There are, and I didn't go into that, but there certainly are particular areas of the brain that appear to be more affected in autism. But when we look and we see, well, this patient has autism, solely autism, and is very high functioning, and then we have a change in the same gene that leads to a patient having autism and significant intellectual disability, that is very confusing and confounding for us. It is true, different genes are more important in different areas of the brain, but I don't think we know enough yet to be able to tell that. And remember, when we talk about treatment, we deal with this all the time in lysosomal storage disorders, some of which affect cellular function in the brain. When you start getting treatment, the problem is location is everything. It's like real estate. When we're treating autism, treating cells in your muscle is not what we're after. And we are wonderfully made, and our blood-brain barrier likes to prevent things from crossing into our brain to affect treatment. So there is another obstacle to treatment, and we're going to have to not only design drugs that treat particular groups with a certain, let's call it, category of autism, we're also going to have to design drugs that can get across that blood-brain barrier, which typically means they're going to have to be fairly small molecules to get across and to be able to disperse. And some parts of the brain, we've learned, are more difficult to reach than other parts of the brain with medication. So all of that comes into play when you're trying to design a treatment.
So, Dr. Rogers, thank you. You said that there are others that know more about the genetics of autism than you, but I doubt there are many that can present so clearly on such a complex topic. And I love, having lived through years of headlines of, gene for autism has been discovered. I think that your take-home message of the complexity and the need for subtyping and stratifications is so true. One practical question, for those of us who may be working with families and ordering genetic testing, they're not ordering or referring to you as well. With whole exome sequencing, I guess two questions. One is the cost and the barriers in that regard, and the other is, in what percentage of children with autism who have whole exome sequencing would you estimate we might find an etiological?
I think the numbers would tell us in 10 to 20%, we might find an etiology. Now, that doesn't mean you have a treatment for that etiology, but as we know, many times parents want to know why. And once they get past that, they are better able to negotiate that psychological hurdle of getting the services that their child needs. So 10 to 20% there. I also must tell our audience that the more of those DNA base pairs we sequence, the more likely we are to find benign changes in genes. And that is the key. We have, if you saw behind the scenes, when I'm talking to a patient, if you went to our lab, you would see them with multiple computers and software and sorting through an incredible number of variants, many of which may be benign, but for which we do not have adequate information yet. So if you do gene sequencing, you're going to get changes that are benign, likely benign, variants of uncertain significance, likely pathogenic and pathogenic, we rely on our software and our incredible PhDs to be able to sort through those, and then for us to take the information they give that, combine it with the clinical information, and come up and say, gee, this is the gene that's causing the problem. And there are times when, based on their strict criteria in the lab, because every lab is supposed to do this the same way, they will say, well, that's still a variant of uncertain significance in this gene. I'm looking at the patient, and I know that gene is associated with the syndrome that that patient has. And I will say, I don't care what you have to say. I am positive that this gene is what's causing this child's problems. So it goes both ways. They suggest things and we tell them things, they tell us things, but it takes an incredible team to do molecular testing.
Thank you. And the importance of genetic counselors as well when it comes to.
Oh, absolutely. Well, this is very complicated information, and to be able to present that in an informative and understandable format to a family requires certainly a certain talent and skill to be able to get that across. I always tell people I'm blessed that I grew up on a farm, because I'm a country boy. So I like things simple. So I can talk it up or talk it down, but.
Thank you. I'm cognizant that we're a little over on time. One last question from me, and I'm being selfish. What do you think is the timeline for when whole genome sequencing testing might be more generally?
I think, and right now we have to kind of skirt around that. We do what I'll call pretty good genome sequencing at our lab in Greenwood now. But getting that paid for is indeed difficult, because insurance companies, hope they're not on here, make money by denying payment for services and holding on to their money that their payees give them in their premiums. So it takes a while for us to get a test through, but I do anticipate in the next 1 to 3 years that whole genome sequencing will be recognized as the go-to test. Now, that may obviate the need for old dinosaurs like me, who still like to diagnose patients looking at their features. But as long as we can help the patient, I don't care whether I'm obsolete or not.
Great. Well, thank you very much. You do pretty well for a country boy. So I would just like to thank everybody for joining us today. Once again, if you're claiming SC Endeavor's credit, please make sure you complete that Google form, and that is still in the chat. And we will be providing a recording of this session to all attendees via email in the coming days. So thank you very much, Dr. Rogers. I see Dr. Whitaker had put a note and saying she attended many talks on genetics of ASD. This is one of the best, and I would fully agree. So thank you. And
well, thank you very much.
And thanks to all of our participants. Be safe and enjoy the rest of Friday and the weekend, and we'll see you next year. And do complete the surveys for us. Thanks.